throughout their life. So in order to help the animal to adapt to this daily change, all the organisms, even the plant, microbe, they develop this internal clock system to keep track of the time and help them anticipate the upcoming change of the environment to help them better survive in the wild. And this is not only in animal, in the fungi and all different organisms. And through evolution, they probably have the parallel evolution to, but they all come up with this circadian clock system. It's quite interesting. And one of the most important environmental change is daylight cycle. Just I mentioned, during the day there's bright sunlight. During the night there's no light, no sun at all. This is the picture of the NTU campus. This on the left side is the NTU campus, the image taken during the daytime. On the right side is an image taken during the nighttime. Can anyone guess that what is the light intensity different during this daytime and nighttime? And anyone in the NTU, can any of you guess the light intensity difference during the nighttime and daytime? Any numbers? Or anyone at the Japan want to guess what is the difference of our environmental light brightness different during the daytime and nighttime? Any number? Or anyone? Or you can 10,000. Someone say 10,000. Oh, it's much more than 10,000. Okay, it's 9 logs difference for the light intensity between the daytime and the nighttime. And every organism living on the surface of the earth, they have to accommodate this huge difference. So by this, you would probably think, okay, yeah, the circadian rhythm probably is important. So that's why every single all the organism has this circadian time system. And the starting for circadian time is quite relatively young field in the science, but through many literature, there's a lot of hints for that our physiological function are under control of the circadian clock. Some of you probably know this graph. This is acupuncture book from the early Chinese medicine. It's about, I think through the history, there's about 1,200 years old book. Okay, so this is a book saying that in our body, although some of them are not really cannot be proved by science, but the basic idea is that it shows that our body, our organ, our system, our organ system are working at different times. So through different time, probably your liver are more active than your lungs, and the kidney are most specifically activated during the nighttime. And but although this is require more scientific confirmation, but at least I think this is probably we can find the earliest document showing that we actually have some kind of physiological fluctuation that throughout the day to accommodate this daylight cycle. And of course, this is more recently science discovery showing that, yeah, indeed, our physiological function throughout the day is not the same through these 24 hours. We have higher alertness during the daytime. Our best muscle coordination are around afternoon, around 2:30 p.m. And our body temperature dropped after the sunset, and it will rise just before the sun bath, before you wake up. So if you think about this, our body, our physiological function are actually trying to help us anticipate this our activity and to match our activity best fit to the environmental light out cycle. And also our hormone secretion are under circadian control. Here is a review in 2001. They show that this is from a human data. They put a human in a constant condition. They put a human tester in a constant room that has a constant temperature, constant lightness, and that person sitting in a chair, and they ask him, her to stay awake for this whole time. And they take their blood and they measure like the melatonin is a hormone release. So it go up during the nighttime and it drops after passing the midnight. And the core body temperature also fluctuates. So although we think our body temperature are constant, the same, no, actually our body temperature have this plus 1 or 2 degree of the fluctuation. It will rise before you wake up and it will drop before you go to sleep. And also the reaction time is fluctuates. So that means our nerve system also have the circadian rhythm that is you have sometimes your nervous system is faster, your response faster than some other time. So this is just the case showing you that our physiological function is not exactly the same throughout the whole day. Even though if you just don't go to sleep, this is not sleep induced. Okay, so even if you don't go to sleep, you stay awake for more than 24 hours, this kind of physiological rhythm would still fluctuate. And of course, now we know it's controlled by our circadian clock system. So what else is the human only species that have circadian rhythm? Of course not. If you only look at the very, very simple organism, they also have circadian clock. This circadian rhythm that's shown with their physiological function. So what the organism, the simple organism like the bacteria, we also know that there's perhaps they have this daily cycle of their physiological function. Many of you probably working in the lab. So what is the lab organism you use to do the cloning? I think everyone probably do molecular cloning, right? Even in the lecture, in the undergrad laboratory course, you're probably doing some cloning. So we use E. coli. And this is the graph. This is a very early experiment. At that time, the scientists haven't know that this is actually under control of the circadian clock. But they inoculate E. coli in a very, very long glass tube filled with the media for them to grow. And if you have the experience growing E. coli, how do you know the E. coli is growing more or less? Can anyone tell me? Or when you are growing E. coli, if you take a tube and look at the tube that you are growing this bacteria, how do you know, okay, this tube has E. coli growing in there, this tube doesn't have it? Can you just look at it by eye or by any measurement? What do you measure how many bacteria are in the culture media? Anyone? No one ever grow E. coli in the lab? See the colors? Yes, exactly. So you can just look at the liquid's color. If you have more bacteria growing in the liquids, you will become less transparent. So many of you probably testing the E. coli growing concentration by using the OD. So you put the light through and see how much the light is absorbed. The more absorption, that means there are more bacteria in the culture medium. So they do this, okay? They put the E. coli in a very, very long glass tube, and they look through the growing in the long tube, and then look at their density, so they can be converted back to the growing rate. And you can see it here. This is throughout the time, okay? This is hours. This is growing rate. You can see here the growing rate increase, dropped, increase, drop, increase, drop, increase, drop. You can see this regularly patterns, rhythmicity pattern of their growing rates. This is just a very simple observation that even the bacteria have this circadian daily cycle. Their growing rate peak once every about 20 to 20 hours, very close to 24 hours. So this is an example that even the bacteria have this rhythmicity, the growing rate. Of course, we can do a better experiment nowadays after we know there is a circadian clock. So scientists also do this. They put the bacteria on an agar plate. So they put this dot the bacteria on the agar plate, and they will grow and to a ring-like structure like this. And if they put some dye in the media, when the bacteria are growing faster, then they will uptake lesser dye accumulate in the bacteria. This is if they grow slower, they will uptake more dye in the bacteria. This is very similar to the tree ring that you can count them. When they grow slower, they'll be the color will darker. When they grow faster, the color will be lighter. This is exactly the same thing here. And when they put the bacteria on a plate and they measure it, they can find that this darker band appear once per day. So every 24 hours, the dark band appears once per day. Okay, so this is just a quick example to show you that human, as complex as human, or as simple as bacteria, we all have this kind of a daily reason that govern our physiological function, govern our growing and the growth rate of the bacteria. So, but this is just the daily reason. How do we really know? This is a circadian reason. This is something endogenous clock that's helping this organism to adapt, to anticipate the changing of the environment. So there are some criteria for circadian clock. Okay, so the first one is, of course, because this is keeping track of one day. Okay, you have this environment cycle once per day. So this cycle, this rhythmicity should be approximately 24 hours. If you have something like 15 hours, it probably is not tracking the day. Okay, so this rhythm should be approximately 24 hours. And the most important thing is because it will help us anticipate the change of the environment, it needs to be endogenously rhythmicity. Okay, so that means even in the constant condition, if you don't give the individual any hints, any cue, any environmental cue, this rhythm should be still keep going. Okay, so under the constant condition. And the second thing is we need to have this rhythm be adjustable. Okay, if this is an endogenous rhythm, it just keep running by itself, then it have no use at all. The animal will move around on the land, and you will probably happen to move across the time. If the clock cannot be adjusted, it's just really hard for animal to use it. So this clock should be able to adjust. That means the environmental signal can reset the clock. The clock should be able to match the external 24 hours light dark cycle. We call them entrainment. Okay, so the clock should be entrainable. That means the clock should be able to be matched to the external 24 hours. And the last one is the temperature compensation. I will talk about more detail later when I give you some example. So I'll give you some example to talk about these three criteria. The first one is this reciprocity approximately 24 hours in a constant condition. We call it free run. So it's like the clock. It doesn't forced by any external cue. The clock just run by itself. We call it free run. Okay. Here is an example of a human free run. So this is Michel Siffry. Is a French archaeologist. So his job is go underground and study the underground cave. Okay, and he loved that, and he needs to go underground. In the underground cave, you can imagine that it will be completely dark, so no light dark cycle. In the underground cave, the humidity is keep constant. It's about 99% humidity all the time. It's very, very humid. And there's no temperature fluctuation because it's underground. There's almost no temperature fluctuation. So for him to go underground, it's just happened to be a constant environment for him. And he finds some interesting thing. Once he go underground to do the study, he often go there for months, 2 months, even the longer time he go 3 or 4 months underground. And he find that when he asks his assistant to give him food from the entrance, his assistant find that every time he call up, every day, he's call up like 15, 30 minutes later than the previous day. And it's quite constant. So one time when you go underground study, he switched from an archaeologist to become a biologist. Okay, trying to get our job. Okay, this. He put a lot of sensor on him, and he measure his sleep time. Okay, so here is the graph of his sleep time. This is day. Each line mean a day. This is day 1, 2, 3, 4, and the next line is day 2, and day 3, day 4, day 5. So and the black bar mean when he is asleep. Okay, so you can see that at the beginning it's probably not really clear, but here after a couple weeks you can see his sleep time is really it's consistently showing through this time, and it's later and later every day. But you can see it's not randomly. He does not sleep randomly, even though he say after in the underground after maybe 1 week or 2 weeks, he completely lost the conscious about the passing of the time. So he don't know anything. He cannot keep track of the time consciously, but his sleep wake cycle still remain very beautifully. And one thing very interesting is this is you can see that this is time every day he sleep later and later. Okay, this is beautifully slower every day. So if you look at this, you can calculate the sleep time for him every day, and you will see that for this, when there's no external light dark cycle, his internal days, his internal clocks decide each day is from this time when he sleep to the next time when he sleep is about more than 24 hours, right? So for human, our external clock is about 24.3 hours. That's really interesting. That's why all of us cannot get up during the morning. Our clock is 24.3 hours. That means our clock runs later and later and later every day. It's about 15 to half hour later. So next time when you're late to the class, you probably told your teacher that, oh, sorry, my clock is free running. Okay, so you can have a scientific excuse to be late in the in the classroom. So this is for the human. And for the nocturnal animal, this is an example for a flying squirrel. This is a nocturnal animal. Also, when we keep this animal in a lab condition for constant condition, their circadian clock, this is the activity reason. They stopped wake up at this time. And you can see that it wake up earlier every day. For most of the diurnal animal, we have our free running periods longer than 24 hours. And for most of the nocturnal animal, they have their free running periods less than 24 hours. So there's almost no animal have their circadian period, free running period, exactly 24 hours. So I will explain this a little bit later, but there's almost no animal have the exactly 24 hours circadian clock. One thing is because animal, they will see the sunlight, so their clock can be adjusted by the external light dark cycle. So they don't need to have a 24-hour clock. But for some animal, very special case like the insect, when they are in a pupal stage, they cannot move. A lot of time they're buried underground or inside the tree. At that time, during that time, they cannot sense the external light dark cycle. So some very few example for especially for the insects during the pupa stage, their endogenous clock can be exactly 24 hours. But for most animal, the free running periods are not 24 hours. And for the second criteria, the endogenous clock should be able to match the external light dark cycle. We call it photo entrainment. Okay, this word photo entrainment. The entrainment means matching the endogenous clock to the external light dark cycle. So we usually use a mouse as a laboratory animal to study circadian clock, because if you have any experiment have mouse rodent, you know that they really like to run on the wheel. That's what they do. They really like it. So as scientists, this is an easy way to measure whether they are awake or asleep. If we put a wheel in their cage, when they're awake, they're just so happy to see the wheel and then just keep running on the wheel. So we can just record whether the wheel has been run by the animal, and then we can put the bar graph here. The black dots mean they are running on the wheel. If we put the animal in a 12-hour light dark cycle, the mouse is a nocturnal animal. They will run, so you will see the black dots during the nighttime. If we move the light dark cycle earlier, like 6 hours earlier, like if you go to Hawaii for a vacation, there's a 6-hour time difference. For the first couple days, you feel jet lag, right? You cannot eat well, you don't sleep well, and your body clock are not synchronized to external light dark cycle. But after a couple of days, your circadian clock readjusts. So, but it takes just a couple of days for your circadian clock to be readjusted. We call it photo entrainment. So basically, your clock are slowly resetting to match the newer light dark cycle. And the same thing is after you come back, your clock needs to again reset to the external light dark cycle. So it's not jumping. Our clock cannot directly jump many hours. And that's important because for the endogenous clock, you want the clock to be quite stable. So even sometimes you have happened to have some weird external inputs to your circadian clock, you only change a little bit, probably thinking this is not normal, this is not right. But if that external signal keep coming in to ask you to reset the clock, then your clock will be reset. But the scientists are still trying to figure out why our clock is resetting so slow. But evolutional time point is because we have the jet plane right now. You can travel around the world very fast, but for normal animal, you never travel in this distance. So the clock doesn't have to be changed so fast. Okay, so we can be in trend, and this speed is depend on when the clock reset is the speed of the resetting, and the direction of the resetting is depend on when the light is perceived by the animal. So during the daytime, there's a dead zone that the light cannot reset the clock, and this is really easy to understand. During the daytime, especially, this is during the noon time. Usually, you will expect that there's bright sunshine during the noon. Okay, so our body, our clock, know they expect this time should have the bright light, big light input. So at this time, no matter how many lights you give the animal, you won't change the clock. But outside of the dead zone, then you can change the clock, especially during the early nighttime. When you shine the light to the animal, you will think, no, it's not right. It must not be a nighttime because there's still light there. It should be a daytime, right? So it will delay its clock. Okay, and during the late night, if you shine the light to the animal, you will think, no, it should be already morning, so he needs to readjust his clock faster to advance his clock. So this means the clock can be setting to different direction during a different time of the day. Through this combination, then we can reset our clock to whatever light or cycle you are experiencing. And because of this resetting time, sometimes if you get the light exposure at this time and get the light exposure later during the nighttime and also early during the nighttime, it will be canceled out. And so for you, this is a quite good thing for you to know that there's actually an iPhone app tell you this. We already have this response for human, and to tell you that when you should, if you go travel internationally to different time zones, then you can calculate which time will give you the best resetting clock. And you will want to go reset the clock in one direction. You will probably want to, like, if you want to adjust your clock later, then you probably want to expose to light during the subjective nighttime, early nighttime, and avoid the light during the late nighttime. And in that way, it will accelerate your resetting time, how many days you need to counter the jet lag problem. And the final one is temperature compensation. Okay, so the temperature compensation means that the clocks are running at very, very similar speeds, no matter the temperature is. If you know the chemistry, like the chemical reaction, usually it depends on the energy balance of the chemical reaction. For the higher temperature, for typical biological biochemical reaction, the higher temperature means faster reaction time, and the lower temperature means lower, slower reaction time. And usually the case is if you take the reaction time at a specific temperature T and another reaction time during a temperature 10 degrees Celsius higher than temperature T, and this reaction usually go 2 to 3 times faster. Okay, so the higher temperature, the biochemical reaction are faster. But for the circadian clocks, you really don't want a faster clock when the temperature is higher, and slower clock when temperature is lower. The Earth doesn't change their rotation speeds during winter time and summertime. It's always 1 day, 24 hours per day. So our circadian clocks need to compensate for this typical biochemical reactions speeds. So the circadian clock, the Q time is approximately 1. So even if you give this 5-10 degree increase in temperature, the speed is the same. The one example scientists already know is coral reef in the ocean. Okay, so in the ocean, although the temperature will not change like 10 degree, that's high, but it will change probably 5 or 6 degree. That means the biochemical reaction time will increase at least 150% if you give this from 24 degrees Celsius to 30 degrees Celsius. What does that mean? That means if you have 24-hour clocks, 150% increase by increasing 5 degrees, you will have your clock running at 16 hours per day. That's 8 hours faster. You don't really want that. So there's a temperature compensation. So if we incubate the coral reef in the different temperature, you can see that this is when they release oxygen, because there's a symbiotic organism living in them will release oxygen. It will release oxygen during the daytime, and smaller oxygen release, lower oxygen release during the nighttime. And in the 24 degree and in the 30 degree, it's the same thing. The speeds are the same. In the constant condition on the right side here, if you don't give them any light dark cycle, they still show this free run period, right? I told you before. And this is the 24 degree, this is 30 degree. The peak time locates exactly at the same time, meaning the clocks run at the same speed. So this is a temperature compensation. So combine 3 together, then we know this. This definitely is an endogenous time keeping clock in our body that tell us that, okay, now it's time to wake up, or which function you need to activate your body. So where is this clock? Actually, this clock is in our body, all the place. You have the clock in your liver, you have clock in your nervous system, you have a clock in your epithelium, you have a clock in your heart, so in your eye and your muscle. So all body parts have the clock in them, but all this clock need to be synchronized together. I think that is most important information. Clock needs to be synchronized. If you have clock all over your body and they all run by themselves, then your body don't have this synchronization, a coherence physiological function. So the main thing is all this body clock need to be synchronized. And who synchronized this clock? This is an early example of the study that show who synchronized on the clock. This is from an insect. On the left is moth, this species of moth. On the right side is another species. And this is interesting because for them, their pupil will close during specific time. On the left side, this is daytime, this is nighttime. They usually close during the early daytime. For many insects, the eclosion will be either very, very early morning, or on the right side, during the evening time, just before the sun sets. If they take out their brain, okay, the eclosion time becomes randomly just distribution in the full whole 24 hours. This indicates that the neuron, the primary master clock that's synchronize the whole body is located in the brain. Even if they remove the brain, this is only can be do in the insect. They put the brain of this left moth onto the right insect, the abdomen. So they have the brain transform. So they stick the brain on the left animal to the right and the right inside to the left, and their eclosion time become reversed. So this just show a very good example to show that the brain is the primary master manipulator to control the whole body clock. And for the mammal, it of course is in the brain, and in the nucleus we call it suprachiasmatic nucleus, SCN, in the hypothalamus. Okay, I just want to show you quickly that if we take the SCN out, this nucleus in the brain, and put it in the culture, if it have the ability to keep running as long as you keep them alive in the culture system. If we look at their, if we put the fluorescence in this cell, and this is of course under control of the clock gene, and you will show this beautifully 24 hours rhythm. It's just become brighter and dimmer once every day, and this is quite extraordinary in the SCM because in the brain they have a very strong power to synchronize to each other, and thus give them the power to control the whole body clock by this neuron system. Finally, the final part is how this circadian clock generates. Okay, so in this, in all the cell, we have this transcription and transcriptional feedback loop and translation modification to accommodate for this endogenous circadian clock. So here is an example from an insect clock. Okay, so for a clock to oscillate, the easy way is you have a positive element and you have the negative element, and this negative element can have the feedback loop onto the positive element, and the positive element can activate the negative element. By this just two elements, then you can have the rhythmicity being generated. So in Drosophila, there's two main protein. They are positive element. We call it cycle and clock protein. They are transcriptional factor. And there are two negative element. We call it periods and timeless. Here is per and tim. And clock is clock. This cycle is CYC. And what they do is the clock and cycle will form heterodimers and binding to the genome. The binding site we call it E-box. And the per and tim and other clock control gene have this E-box in their promoter region. So that means the clock and cycle control the expression of per and tim. Okay, and the per and tim, after they've been transcribed, and they will be translated into the protein. And the protein, the per and tim protein, will form the heterodimers and then go back into the nucleus to inhibit clock and cycle. And so that will form a loop. After inhibition, then the clock and cycle can no longer activate the expression of per and tim. And after you inhibit the per and tim transcription, and after a while the protein, per and tim protein be degraded, then the inhibition is lost, is removed. Then the clock and cycle can again transcription, start the transcription of the per and tim, and then the next cycle begins. So just by this simple transcription feedback loop, then you can generate this daily rhythm, this rhythmicity, constant rhythmicity through almost all the cells in your body. But one thing is really important is just by this transcriptional feedback loop, it is really hard to generate a 24 hours rhythm. Imagine that you have feedback system, really good feedback system. The per and tim will inhibit clock and cycle immediately. What will happen if you have the clock and cycle binding to the E-box and you have the transcription translation. Usually protein will come up just minutes after the transcription, and you will go into the nucleus, inhibit the clotting cycle, then immediately your protein level drop to the and then you supposedly never have a really accumulation of the per and tin protein, then you really cannot get this racemicity happen. So the key point is to have this post-translational modification, so you can have per and tin accumulate slower, so they don't immediately go into the nucleus to block the activity of the clock and cycle. So then after some time delay, they go in to block the clock and cycle. So then you get this beautifully daily rhythm. So the post-translation modification is one of the most important, also a very important part. So in addition to transcriptional feedback loop to come with this positive element and negative element, the post-translation modification is also important because it provides the time delay to let the negative element have the chance to accumulate. So I think you all learn cell biology. So what kind of post-translation modification is there the cell use? Can anyone give me some example? Or anyone from Kyoto, from Japan? What kind of post-translational modification cell can use? Not specifically for the circadian clock, just anything. Can anyone know any kind of post-translational modification?
From Kyoto side, maybe phosphorylation.
Yes, phosphorylation is one of the most common post-translation modifications. That's really good. So anyone from NTU, is there any post-translational modification you can think of? Anyone, use your microphone. Post-translational modification. No one? No? Histone modification. Histone modification, you can have acetylation or methylation. That's also on the histone protein level. That's also kind of a post-translational modification. So a protein in the cell can be modified after they after translation. So for per and tin protein, their post-translation modification is important. And what they use phosphorylation. So here is how the clocks, the molecular clock happen throughout the 24 hours. Okay, so during the midday, the clock and cycle will form heterodimer binding to the E box and transcribe per and tin. The protein of per will be translated and it will be phosphorylated very quickly, phosphorylated, very just single phosphorylation. This phosphorylation will lead to the degradation of the per protein. Okay, so this is the primary mean for the time delay. So when there is just a little amount of per mRNA in the cytosol and you translate a little amount of a per protein, it will be simple phosphorylation, one phosphorylation, and the per protein will be degraded. So it cannot immediately go back to the nucleus to inhibit the clot and cycle protein. So in this case, the per mRNA have the ability to accumulate throughout the time. Okay, and after that, after the accumulation of the mRNA and the per protein being kept degraded, finally the mRNA become accumulate into higher amounts. Then you can have more per protein being translated. Although there's still simple phosphorylation, then in this case, the timeless protein, will bind to per and help the per be even more phosphorylation. So more than one phosphorylation. It will be phosphorylate multiple sites in the per protein. and this multiple higher number of the phosphorylation will prevent PER being degraded. Okay, so this is showing just saying that at the beginning, when the PER is just single phosphorylation, you'll be degraded. When it has more than one phosphorylation, then you will not be degraded anymore. And then this PER protein can also start to accumulate during the evening time. And finally, during midnight, the PER protein and timeless protein will also form a heterodimers and then go into the nucleus to inhibit the activity of the clock and cycle, and finally blocking the transcription of the PER and TIM. Okay, so then at this time, there's no transcription. The mRNA of PER and TIM start to decrease. Of course, the PER and TIM protein translation will be reduced, and the PER and TIM protein will be even hyper phosphorylation. That means, okay, they're being degraded again. So this is really interesting. For a PER protein, the phosphorylation have the 3 different stages. Single phosphorylation means degradation. Okay, multiple phosphorylation means, okay, they are stable. And finally, hyperphosphorylation, all the sites being phosphorylated, they will be degraded again in the nucleus. And after they've been degraded, the clock and cycle can again binding to the E box. We go back to the same cycle to the midday. The clock and cycle can finally again binding to the E box and start the transcription of the PER and TIM mRNA, and then this go through once every day and to form this circadian rhythm. So if I'm telling you that this clock, this is transcription factor, so and all the cells in the body have this clock, then how it's control our physiological function. So later, our presentation, the student presentation, I pick a paper showing that all these clock transcription factor have multiple binding site in our genome, and we'll know how this clock can actually control our physiological function. Although there are just couple, now we know there are probably 10-ish this clock protein, this circadian cycle protein. But this 10-ish protein can control our whole body, all the cell, just by a huge amount of ability for them to bind to the genome and to influence the normal transcription in the cell. And that we will discuss during the student presentation. And I think because the time, I still have 5 minutes. So in the last 5 minutes, I will tell you a little bit about, so now we know there's a clock everywhere in our body. And the SCN is the master clock that synchronizes all this little clock in our body. And every small clock have this transcriptional, translational feedback loop to control the oscillation. And in the student discussion, we'll tell you how this can interpret, this can affect our physiological function. And then I told you the light-dark cycle is important. So how does the light-dark cycle convey the information to control our clock? So in the retina, we have 3 different kinds of photoreceptor. We have the classic photoreceptor, rod cell and cone cell. They are important for us to see, to perceive our environment. They are important for visual vision, pattern vision function. And there's another cell, I'll skip that. We call it IPRGC, intrinsically photosensitive retinal ganglion cell. It's shown here in purple. I don't think you need to know that. You just need to know that in the retina, there is a 3rd photoreceptor. We call it IPRGC. And this IPRGC, they're being activated best by the blue light. It's about 460 nanometer of the blue light. The 460 nanometer blue light is located at this range. Is sky blue, or you can say sea blue. Okay, so it's not very dark, not the purple-ish blue lights. And this cell being activated, it can transmit the information to the SCN, adjust the clock. And this IPRGC is not important for you to see. So in our visual system, we have completely two different systems. One is, you probably learned this in the biological 101 class, that the classic photoreceptor rod and cone, this system is allow you to see. Another system, new system, is IPRGC system in the retina. It's specifically sensing these blue lights to adjust your clocks. So if you during the daytime you get this blue light inputs through the IPRGC, then you can tell your body, your circadian clock that this is daytime. During the nighttime, if you have excess blue light exposure, then you will adjust your clock. So it's really not good to expose to this light during the nighttime. You will readjust your clock. So you're probably thinking about it if you want, if you are exposed to the blue light during the nighttime. So the take-home message is, I want to say is that almost I use a lot of different examples, different organisms in this class to show you that almost all the organisms, from the complex organism like human to simple organisms like bacteria and coral reef and insects, we all have the biological clock inside our body to help us anticipate the change of our environment, to help us survive better. And the second thing is this circadian clock control many important physiological functions like activity, sleep. It affects our hormone release. It affects our immune system. Our immune system actually are active for human. Our immune system are active during the daytime, and then lower during the nighttime. So if you want to go out, probably the daytime is a better time for you to go out. Our immune system will increase their activity during the daytime, and our stem cell activity, especially the skin stem cell activity, will increase their activity during the nighttime, because the sunlight, the UV light, is bad for DNA. So when there's a DNA duplication for the stem cell to divide, it doesn't want the UV light to expose them. So our stem cell, especially the skin stem cell, are active during the nighttime. So a lot of physiological functions are controlled by circadian clock. And finally, the most important thing is the transcriptional translation interlocking feedback loop. So it's not only transcription, it also is not only translation. Combined together, combine transcription and translation together, then we have this core molecular clock in the cell that's generate this 24 hours LiDAR cycle. And so I think this is today's lecture for the circadian clock. We'll take approximately 5 minutes breaks. We'll come back 5 minutes later, and the student will do a paper presentation. But before the break, is there any question for the lecture?
Hello, thank you for the very interesting presentation. So as you mentioned many times, the day-night cycle is very important for determining circadian rhythms. So I was just wondering how, for example, there's some deep sea creatures who are adapted to live in conditions where there's no light at all, how their circadian rhythms are different. Okay. And how the regulation is different.
Yeah, that's a really good question. So you are asking, what about those animals never experience the LiDAR cycle, like the animal living in the deep sea? And also there's animal living in the cave, in the underground cave. And also there's animal living in the Arctic that have constant day during the summertime. And for those animals, I was just talking about the first one is scientists actually looking at the fish living in the underground cave. They don't have eye, their eye degenerates, so because there's almost no light, they don't need the eye. And all those those clock protein are still exist in their cell. and their core clock protein also oscillates, but they are not synchronized oscillation. They no longer interact with each other, so they don't have this 24 hours circadian rhythm for those animals that live in a place that never see the oscillation of the light dark cycle. And this is really important is for the plant, for the bacteria, for the animal, for the fungi, they living on the surface of the earth, they all have this circadian rhythm. It's the molecular clock is not commonly evolved. They are each different lineage have come up with their own circadian clock, but it's just happened that all of them use the transcription translation feedback loop to generate this clock. So it's kind of an evolutional point of view that if you're living on the surface of the Earth, then you need to have this circadian clock. For very few organisms, never see the light out cycle. Their circadian clock protein is there, but they never found this interaction to show the daily reason.
Thank you.
Okay. We have a question from NTU. Okay, so the question is asking about whether the embryo, like in the mother, have the circadian rhythm. We're just talking about this a couple of days ago in my lab. For human, this is the case for human. For human infants, the baby, when they are born, for the first 2 months, they don't have circadian rhythm. When they don't have circadian rhythm, their sleep-wake cycle look like this. I want to come back to here. If you look at the baby for their first 2 months, their sleep-wake cycle is like this. It's randomly. They just sleep anytime they want. They wake up anytime they want. There's no daylight cycle. So that's why all the parents are like sleep deprived for the first couple months. So in the infant stage, there's no circadian rhythm. The protein is there. It's important for development. In some case, like in C. elegans, the circadian clock protein, if you have mutant, then their development will be delayed. But exactly the rule of this protein during development is still, there's still much more to be, should be investigated to figure out what they actually do during development. But for coherence oscillation, like the once per day oscillation, it happens after birth. And for human, it happens for maybe the 3rd or 4th month after birth. So don't train your children for the first 2 months to sleep through the nighttime. They still don't have the clock yet.
That's very interesting. So why so what part is missing?
Okay, so there are 2 things. The first thing is the clock is waiting the input from the external light-dark cycle. So at that time, the connection, the neuron connection is not fully formed yet. So for mammal, this is in the case of a mammal. For mammal, their inputs will connect to the brain, specifically the SCN, the master clock, after birth. So we don't know why it takes such a delay. For visual connection, usually it's being formed during the embryonic stage. Although you cannot see, the baby cannot see very well, but the connection is already there. They need to refine the connection and then modify the connection for better vision. But the connection is already there. But for the circadian clock, we still don't know yet why they are slower. But at least we know that when the animal birds for the first couple of days for the mouse, for the first couple of months for human, the eye inputs to the circadian clock is not connect. The neuron connection is not there yet.
Question from Thank you for the lecture. Now, another student from Kyoto side asked about animals that couldn't feel the light. So I just wonder, can animals keep track the day in any other form, like maybe for animals living in the sea, the tide change?
Yes, this is a really good question. I didn't really go into that because of the time limitation. The light is the most strong environmental cue, but there's not only light. There's many other information can set the clock, like the food. If you keep the food animal in the laboratory and you give them food during the specific time, then you can set their clock. And also the temperature reason can be set for animal, and like in the birds, if you give them daytime 30 degrees and nighttime 20 degrees, their circadian clock can be set by the temperature fluctuation too. And the most important thing is even the social interaction can set the time of the clock. This is they did this in the chicken. In the I don't know whether you really heard the chicken during the morning. They will make the noisy sounds during the morning. And if you play this chicken sound to another chicken, that sound can set the clock for chicken. So there's some social interaction too. So even they hear someone singing or something, they can reset the clock. So the clock can be reset by many environmental cues. But if all of this exists, the primary, the strongest force is light. So if you contradict all other with light, the light usually win. Light and food is complicated, but all other sensory inputs, light is the strongest cue. Okay. If there's no more question, then we will take just maybe 2 minutes breaks. It's slightly late, and which the student will set up the slides, and then we'll start the presentation immediately after they start set up the slides. So we'll just take 2 minutes break. Okay.
Good morning, everyone. Today, my name is Liang Feng, and we are going to present our paper, which is transcriptional architecture and chromatin landscape of the crow circadian clocking mammals. The circadian clock in mammals is cell-independent and is composed of an autoregulator transcriptional network with interlock feedback loops. Although the majority of core components of the circadian gene network are likely known, and many thousands of transcripts have been shown to express circadian oscillations in various tissues. However, the genome-wide architecture of the transcriptional network regulated by the core circadian clock remains to be defined. And so the paper focused on this gene. So they use ChIP-seq to locate DNA binding sites for BMO1 clock and PAS2, PER1, PER2, and CRY1, CRY2 in vivo in mouse liver. For each transcription factor, 6 time points every 4 hours over a circadian cycle are shown beginning at CT0 and ending at CT20. Knockout mice were used as a negative control. So as you can see on the figure A, BMO1 binds in a circadian manner between circadian time 0 and 12 to 3 sites with peak binding phase occurring at CT4 to 8. The heterodimer partner of BMO1, which are clock and MPAS2, also bind in a time-dependent manner similar to BMO1. The repressor PER1, PER2, and CRY2 then bind the same site between CT12 and CT20. In contrast, CRY1 exhibit maximum binding at CT0, which extend the repression phase into the beginning of the next cycle, which when BMO1 clock and MPAS2 occupancy increase again at CT0. On heat map representations of genome-wide peak location analysis show that, shows that the dynamic of binding during the circadian cycle in constant darkness. Each peak in the genome is represented as a horizontal line ordered vertically by signal strength. 6 time points are shown beginning at the CT0 and ending at CT20 from left to right. Knockout mice control is shown at the far right. The number of peaks in the genome is indicated at the bottom. The blue gradient indicates the coverage or signal for all binding sites in the genome. So from their analysis, the genome-wide occupancy rhythm of BMO1 clock and MPAS2 peak at CT6 to 8, whereas PER1, PER2, CRY1, and CRY2 shows binding rhythm that peak at CT15.9, CT17.3, and CT0.4, and CT15.4, respectively. To analyze the overlap of the binding sites of the circadian regulators genome-wide, they compared the 6-way overlap of the factor BMO1 clock and PER1, PER2, CRY1, and CRY2, and show the results in Cholesky diagram. At the core, you can see there's a 1,444 sites in the genome are bound by 606 factor, and of particular interest are the largest number of CRY1, CRY2, and PER2, sites are uniquely bound by each factor and appear to be independent of BMO1 clock site. In order to assess transcriptional readout on a genome level, they use whole transcriptome RNA-seq to measure circadian gene expression in liver over a 48-hour interval sample every 4 hours in constant darkness. At the per2 locus, sense-strand reads are showing the black, and reverse strand reads are showing the red. As normalized average read per 50 million total reads in 10 pp bins. The y-axis scale are 0 to 40 for the positive per2 strand and minus 40 to 0 for the negative per2 strand. So a high amplitude of RNA rhythm can be seen at per2 locus with peak at CT12 and CT36. At the per2 locus, the intron and exon signal both oscillate and their phase are similar. They compare the intron signal as a representation of pre-mRNA expression or nesting transcription and the exon signal as a represent of mRNA expression. The per2 antisense transcription is circadian and antiphases, as you can see on the graphic. And in genome-wide analysis, they detect 1,371 intron and 2,037 exon RNA cycling transcriptions, which are shown as heat map ordered by the phase of RNA expression. And so a comparison of the phase distribution of the intron and exon cycling transcriptions reveals a difference, as you can see on the graph, that the intron cycling transcriptions are clustered and peak at CT 15.1. However, the exon cycling transcriptions have 3 peaks phase and not distributed across all phase. Comparing the gene in the intron and exon cycling transcript classes shows that only 458 genes are in common, and this is set of common gene is enriched for non-circadian clock gene and high amplitude cycling target gene reported previously. The phase of common intron and exon cycling transcripts are correlated, suggesting that transcriptional cycle primarily drive this mRNA reason. And in the intron cycling, exon line cycling class, the first one, the cycling pre-mRNA transcripts are clustered and at the same phase as the overall intron cycling class, but the steady state of mRNA are likely to have long half-life. By contrast, in the intron not cycling exon cycling class, the phase are widely distributed as seen in the overall exon cycling class. And this reason are likely to rise from post-transcriptional regulatory process. And so they use ChIP-seq to with these 2 antibody, which is 8WG16 and 3E835P, to see how RNA polymers 2 work with this. On a genome-wide basic, RNA polymers 2 occupation is circadian with 8WG16 signal peaking at CT14.5, and the other one signal peaking at 0.6. Examination of the time-dependent pattern of CBD occupation reveals that CBP can have either 24-hour or 12-hour binding rhythm. And with the 24-hour binding rhythm is peaking at CT60 to 20, and the 12-hour rhythm peaking at CT4 and CT60. Because CBP has peaked either at CT4 or CT60. This suggests that CBP may be able to function as either a co-activator at CT4 or co-repressor at CT60. In support of this hypothesis, they find that CBP can interact with the repressor PER2 in native expression, expressed in a time-dependent manner, with maxima interaction occurring at CT60 to 20 when the PER2 level are high.
Okay, hello everyone. I'm Cheolun Shen from NTU, and after knowing that the RNA polymers 2 and its co-activator, which is P300 and CBB here, can shows the robust circadian rhythm, and moreover, the CBP can interact with the PER2 protein. We're next going to see that the chromatin state, which is here is the histone modifications, and the RNA polymerase 2's occupancy. Do they also show the circadian rhythms? So in this figure, we can see that the red one is the situation in the RNA polymers 2, and the blue one is the histone modifications. So here we have 4 experimental groups, which is the expressed genes and the unexpressed genes. And so the first 2 groups, we can see that in these figures, the x-axis means that the distance from the transcription start sites, and the y-axis means that it can be seen as a signal intensity in some kind of expression level. So we can see that in the expressed genes group, both the RNA polymerase 2 AWG16 and the serine 5P proteins, and the 3 of the histone modifications, like such as S3K4 trimethylation, and S3K9 acetylation, and S3K27 acetylation, both of them shows robust circadian rhythms. But on the contrast, we can see that in the unexpressed gene groups, all of these kind of modifications do not show circadian rhythms. And as for the intron group, we can separate it as 2 groups, which is the cycling intron group and the non-cycling intron group. So first, in the cycling intron group, we can see that both RNA polymerase 2 occupancy and the histone modifications, they all show circadian modulations. But what is very surprising and unexpectedly is that in the non-cycling intron group, they also show the circadian modulations. And here we will discuss later about this kind of result. So now we are going to see that in the case of the chromatin remodeling, there are we know that there are many kind of histone modifications. So like S3K4 methylation, it is a mark that is characteristic of the enhancer sites and the gene bodies, and it will exhibit a very subtle circadian rhythm in this figure. And this S3K4 trimethylation and S3K9 acetylation and S3K27 acetylation, this kind of modifications, they are all enriched at the promoter site, and in these figures they show a strong circadian rhythm in the occupancy at the TSS. And for the last two modification, S3K36 trimethylation and S3K79 dimethylation, they express low amplitude of circadian modulation. So what is interesting is that here we can see that the first two diagrams, they show the anti-phase rhythm. So their rhythm is like they have the opposite phase. So after seeing through this data, so we can have a conclusion by this kind of the figures. So this is kind of a chromatin landscape figure, which represents the expression level of the core circadian clock in different times. So here we can see there are a series of phases, which is the poised state from the beginning, and in this kind and the repression phase. So in these two phases, we can see that the clock and BMO1 complex is like they are at very low level, and the cryptochrome 1 is repressed. But after the poise and the repression state, now we're entering the activation state. So in this in the activation state, the clock and BMO1 complex expression level goes high, and we can see the P300 recruitment, and also the S3K9 acetylation happens. And next, we are going to have a nascent transcription. And in this stage, it means that we are going to have some kind of initial transcription. And so the transcription is going to start and will produce a lot of RNA. And after the transcription state, we'll now enter in the repression state and go back to a cycle. So this is kind of a landscape that can conclude all the data in this paper. So the final part is the discussion. So these kind of dynamics of the transcription provide very important and novel insights on the RNA polymerase occupancy and many histone modifications. And as for the situation in the non-cycling intron group shows circadian rhythms, it can be explained as that the gene expression per se is like tightly correlated with circadian transcription factor binding rather than the circadian rhythm of the gene. And last but not least is that we see a lot of histone modifications like methylation, trimethylation, or acetylation. But what we do not see is the situation in the deacetylysis and some kind of co-repressor. We have seen P300 and CBP, this kind of co-activator, but we did not see the situation in the co-repressor. So it will be of interest to explore the role of the histone deacetylases and the other kind of co-repressor complex in the circadian regulation. So here is our report, and thank you for listening. So any questions?
Okay, first I will read the paper. So I have a question. Did they include any analysis of like anything that might be used to entrain the cells?
or for example, that they look at, rate the question of HDACs, they look at HDAC inhibitors and see how that would affect the cycle.
Sorry, can you repeat your question again? I'm not.
Okay. I'm just wondering if they, two questions actually.
Okay.
One is, were they looking at free living cells, or were they trying to train them in some way? And the other way, another question, try seeing what the effects of an HDAC inhibitor might be.
So they use the free running.
It might be fun to do a thought experiment. What would happen if you gave the animals some jet lag? And the rhythm was kind of discombobulated for a while. Yeah, okay. Thank you very much, Hugo.